Hey guys, so today I decided that I was going to try to make a little study with me video, or watch me study, whatever you want to call it. Sorry, this one little hair is driving me crazy. I am studying neuroscience, and this module we are studying the eye and like different processing of light and things like that, and the neural response to light changes within the eye. So that's what I'm studying, but I just finished actually reading this chapter about the eye, and I have to do some homework. I have to do the chapter questions, and I also have to do the lectures for this module. So I figured that I could kind of let you guys see how I study my neuroscience. This is kind of a smaller module. I think it's because of how long this chapter is and how like in depth you go with the eye, because this chapter itself started on page 293 and goes to page 330. So it was a very long chapter, but like I said, there's a lot of information that goes into the eyeball and the processing of light by the brain and the neural reactions to different changes in light. So if you guys are interested, feel free to watch. If not, then I guess I will see you in the next one. But yeah, I'm going to probably start with the lectures and then do the little homework questions. So hopefully you guys enjoy this little study neuroscience with me.
Which has to do with how we detect energy in the electromagnetic spectrum, and there are certain forms of that light, like ultraviolet light and infrared light, that we don't see. So the question is, why do we just see the range of visible light that we are able to see? And there's no real answer to that, but it's speculative. But this diagram, so this plot shows the amount of energy that's emitted from the sun at different frequencies or different wavelengths on the x-axis, and you can see that it's not uniform. So the sun emits more energy at some wavelengths than others, and then our visible light spectrum is right near the point of maximum amount of light, or maximum amplitude for these different. Some of the physics that varies across different object types, right? So different objects reflect more or less uniformly with different amounts of scatter. Different objects absorb wavelengths at different frequencies, giving them different color. And then there are even some objects that light can pass through. It's just they have different densities in the air, and so you get this refraction. And these are all just the nature of the way light comes from some source through whatever objects exist in your world in order to arrive at your eye. And then your job as an owner of an eye and a brain is to take whatever light arrives at your eye and make sense in some reasonable way of what objects are out there in the world. You'd hope there'd be consistency, so if the light source changes, you still see the same object, that you could make sense. You could, if things are refracted, that you recognize that the degree to which light scatters off objects makes sense, that you can infer color and so on, right? And so this is really what we're going to be dealing with, is how is it that this incoming light is transformed and made sense of to allow us to get a coherent view of what's going on in our visual world. Neural stuff, in this case, different amounts of glutamate release. Okay, so the way this works is through a G protein coupled light sensitive channel. Okay, and we've talked about G protein receptors, second messenger cascades, and so you should have sort of a general understanding of what's involved when you have a G protein coupled receptor. In this case, what happens is light hits what's called an opsin, a type of molecule called an opsin, which is G protein coupled, and when light hits this opsin, in this diagram it's rhodopsin, which exists in one type of photoreceptors, but when light hits the rhodopsin, it causes the G protein to become activated. And the G protein activates and interacts with another molecule. We talked about adenylyl cyclase before with the norepinephrine receptors. Now we're talking about this phosphodiesterase. And the phosphodiesterase is an enzyme that converts cyclic GMP into non-active GMP. Okay, so what does it mean to be non-active? Well, cyclic GMP in photoreceptors acts on ion channels to open them up. Okay, so if there's a lot of cyclic GMP around, then there's a lot of ion channels open. These ion channels are permeable to cations, sodium and calcium. And so when these channels are open, then you get depolarization of the photoreceptor and consequent release of glutamate in proportion to how depolarized the photoreceptor is. And that's so the reason why that's true is because of the light absorption characteristics of the different opsins within these cones. All right, so these are three plots. So on the x-axis is the wavelength of light, and the y-axis is how much each cone absorbs light at these different wavelengths. And so you can see blue cones absorb kind of over a broad range of light wavelengths, but they absorb most intensely right above 400 nanometers of wavelength, which is right at 430 nanometers. So they are called blue cones because that 430 nanometers is blue. Okay, and then their green and red cones are shown in the other two lines. You can notice that their absorption spectra are pretty close, but nonetheless, the red cones are shifted to longer wavelengths than the green, so they're called red and green, again, based on the maximum wavelength at which they absorb. Okay, so light comes in, it hits these three different types of cones, and then based on the distribution of wavelengths in light, you're going to activate these cones to more or lesser degree based on the correspondence between the wavelengths of the incoming light and the absorption spectra of the different cone opsins.
So I hope you guys found that at least a little bit interesting. I tried to share some of the lecture at the points that I thought you guys might find a little interesting, or information that was kind of cool. So I am about to go take my quiz for this chapter, the chapter 9 quiz, but I cannot have that on camera, because I could actually get in really big trouble for showing any quiz questions or things like that. So I'm not going to be able to have that on camera, but I will come in and let you guys know, hopefully, that I passed. We shall see. So I'll be back shortly. So I just took my quiz and I got 100%. That makes me happy. I was going to go to the lake today, but I don't know if you can tell, but outside it's kind of poopy. Gloomy and rainy and all that. So I decided, since we're not really going to be able to do anything outside, then I might as well just stay home and get ahead on some homework, because even though I'm done with everything I can do currently for the neuroscience class, as far as like my reading assignment and the chapter 9 quiz, I can't take the exam until like Wednesday night. I think it opens. So I can't do that now. But I do have my other class, which is the homicide and serial killer class. I do have a reading assignment, a quiz, and a paper to write for that. And then I also have been working ahead on my 2 English classes that I'm taking through the Universal Learning System. And so I know I have papers to write for those 2 classes. So I figured, since it's gloomy and there's really nothing else going on, I might as well take advantage of the bad weather and get ahead. Or get ahead as much as I can. So with that being said, that's going to be the end of today's video. If you guys enjoyed this video, please let me know. If you didn't enjoy this video, please let me know that as well, and I will try not to post any more videos like that. I just figured that sometimes it's nice to kind of see how other people study and what other people are up to. And since I was studying anyway, I just figured I would share that with you guys. But with that being said, that's all I have for this video. I hope you guys are having a great day, and I will talk to you guys again next time.